Hello everyone, welcome to our channel Fox Science. Today we will discuss about our part 2 of translation, and today we will learn about the structure and function of tRNA, as tRNA is the very essential part of translation process along with mRNA and ribosome. So today we will learn how it's structured and how it function in translation process. So let's get started. So basically tRNA looks like a clove. Okay, here in my screen you can see it's a clove like structure. This is the prokaryotic tRNA and this is the eukaryotic tRNA. So how they are different, means prokaryotic tRNA and eukaryotic tRNA, how they differ, we will discuss about after some time. So first we will learn how its structure, proper structure. Actually, so here you can see this is we called acceptor arm of tRNA. Okay, this is we called T psi C arm of tRNA. This arm we called DHU loop, and this is a single V like structure. Okay, so this is the variable arm, and this portion we called as anticodon loop. Okay, so here how many type of structure we can see in a single tRNA? First, this is the acceptor arm. Second, this is the T psi C loop. This is the DHU loop, and this is the anticodon loop along with the variable arm. So why this is called as acceptor arm? Because it accepts the amino acid. Why this is called anticodon loop? Because the codon which are needed to code for a amino acids are present in the mRNA, obviously, and with the mRNA this portion will get attached. That is why this portion will always carry the anticodon sequence, which will be getting attached with the codon present in the mRNA. Okay, clear? So now we will learn about a very interesting thing, that is the difference between initiated tRNA and general tRNA. So now till now we discussed about the general tRNA. In initiated tRNA there will be presence of five nucleotide in this area. Okay, ACCA, along with one base pairing will be missing here, that is the C1 and A72. This base pairing will be also missing here in case of initiated tRNA. Now you may ask why? Because always the methionine, AUG is the first codon, as we all know. AUG codes for what? Methionine. In case of prokaryotes, the methionine is always formilated, means one formyl group is attached with the methionine. So for that formylation, the absence of this base pair, means the C1, A72, this base pair's absence is needed for incorporating the formyl group. Okay, but in case of eukaryote, you can see the initiated tRNA is also for the methionine. Here also the A1 or U72, this base pair. Here you can see that this base pair is present, but the methionine will be the first amino acid in case of eukaryote as well. But here you can see in case of prokaryote, the C1A72, this base pairing will be missing. So this is the signature of the initiated tRNA. Okay. So now, as we already know about the difference between initiated tRNA and general tRNA, let's discuss something interesting about general tRNA. Basically, general tRNA can be divided into two types, okay? Type 1 tRNA and type 2 tRNA. Type 1 tRNA contains 4 to 5 nucleotide in this variable region, but type 2 tRNA contains 10 to 24 different nucleotides in this variable region. Okay, and as we already known that this anticodon loop contains 5 base pairs and 7 single nucleotide, and the I missed in first part, that is the signature of the anticodon arm is the 3GC base pair. You can see here that 3G, GGG, and in this side you can see CCC. This 3GC base pair is the signature of the anticodon loop. Okay. So in this T-Shi-C loop, it contains 5 base pairs and 7 single nucleotide. The acceptor arm always contains 7 base pair, 4 single nucleotide, and the DHU loop always variable in length. So you may ask that what if something mutation happen in this 4 nucleotide present in the acceptor arm. So what will happen? Then they cannot show affinity towards amino acid. Okay, they will lose their affinity towards any kind of amino acid. That will be called as discriminating nucleotide or discriminated base in tRNA. Here we will learn one more very interesting term, that is iso-accepting tRNA. The term iso means what? Iso means same. Accepting means it will accept. So when with single type of amino acid will be accepted by more than 2 tRNA, then it will be called as iso-accepting tRNA. For example, there is tRNA 1 and tRNA 2, two type of tRNA present. Okay. But when tRNA1 and tRNA2 both will code for only glycine, then that tRNA1 and tRNA2 will be considered as iso-accepting tRNA. Just for your information, I would also like to mention one interesting information, that is codon degeneracy. So what is codon degeneracy? When a single amino acid will be coded by multiple codon, that will be called as codon degeneracy. Here you can see AUU, AUA, AUC, 3 codons can code isoleucine. But if you discuss about a single codon, for example AUC, it can only code isoleucine. It cannot code any other amino acid present in the organism. Okay. So that is basically we called as codon degeneracy. Now we will discuss about the vowel pairing. Vowel pairing also a very interesting factor for translation process. So what happens in the oval pairing? Actually, when the codon on the mRNA and the anticodon of the tRNA join, for example, here we can see this is the anticodon loop, right? So this is CAU will be the anticodon region which will get attached with the codon portion of the mRNA. So in case of the first base pairing, for example, in case of C and in case of G, which will be present in the mRNA, so this base pairing will always follow the Watson and Crick rule. Means A will always get attached with the U, C will always get attached with the G. The middle base also will follow the Watson and Crick rule strictly. But the third, in case of third nucleotide, when these two hydrogen bonded, already hydrogen bonded base pairing happen, then the third base pair is weakly, means there may be flexibility or wobble in their pairing. So in 1966, Francis Crick developed the wobble hypothesis, which proposed that some non-standard pairings of bases could occur at this position. What kind of non-standard pairings? For example, if there is G in the anticodon, there can be presence of U or C in the codon region. If C, so sometimes G in the codon. If A, then sometimes U. If U, then sometimes A or G. If I, then sometimes A and U can be present in the codon region. So this is the wobble hypothesis, and it happens always with the third position of the anticodon loop. Okay. So as we have already learned about the structure of the tRNA, now we will know that how it functions. So the function of tRNA is the transfer of amino acid towards the uh, polymeric chain, means to form the polymer of amino acid. That is the main function of tRNA. So for that, tRNA must and should accept the specific amino acid from the environment, then it have to transfer. So how it accept the amino acid from the environment? So this process is called tRNA charging. This process is uh, assisted by one specific enzyme that is called amino acyl tRNA synthetase. This is that enzyme, okay? So what happened first? You can see here the amino acid along with the ATP gets attached with the amino acyl tRNA synthetase enzyme at the active site, followed by discharge of 2-phosphate molecule. So now here only the amino acid along with AMP gets attached with the active site of the amino acyl tRNA synthetase enzyme. After that, what happens? Then the tRNA comes. First, the T-C loop gets attached with the enzyme. Then the anticodon loop gets attached with the enzyme, and finally the acceptor arm, the 3' region ACCA, that 4 free nucleotide accept the specific amino acid. So this is how amino acid gets attached with the tRNA and one tRNA becomes activate. In this slide you will get to understand very clearly. You can see here this is the amino acid and this is the ATP molecule. So first the ATP molecule and the amino acid gets attached, followed by the discharge of 2-phosphate molecule. So now this is AMP and the amino acid. These two attached by a ether linkage, means the amino acid and AMP gets bind with the ether linkage. After that, then the tRNA comes, and this is the acceptor arm of the tRNA, and this oxygen molecule of the amino acid gets directly attached with the tRNA, and the AMP gets dissociated from that compound. Now in this portion you can see this is the ester linkage amino acyl tRNA, and this is the released AMP. So this is how tRNA gets attached with the specific amino acids and become activated. So in this video we learned how tRNA looks like, what is their structure and what is their function, how they become activated. In our next video we will learn about different types of ribosome function in the translation process. If you have any doubt in this video, so please do comment in our comment section. If you like this video, please do like, subscribe our channel and share with your friends. Thank you so much.